Assalamualaikum. My name is Muhammad Hanafi bin Muhammad Samsuddin. My number metric is F21B1138. So I from SBL. So this is the slide for industrial training presentation, Faculty of Agro-Based Industry, July for 2025. So this is the most valuable experience I got during internship. As you can see in the video, I was doing the quality check for the spreading agar with dilution bottle. So I was taught and trained on how to hold the plate properly so there will be no cross contamination while doing the QC process. So this is the one memorable memory that I got while doing my internship at this company will be while celebrating Hari Raya together with all staff working at Arif Efektif. We also play some games while and recorded some TikToks to further strengthen the bonding amongst the staff. It was quite funny and unforgettable experience I got while working in Arif Efektif Sdn Bhd. I would like to express my sincere thanks to Arif Effective Sendirian Berhad for giving me the opportunity to undergo industrial training at their company. I am very grateful to all the staff, especially my supervisor, for their guidance, support, and valuable knowledge throughout the training period. This experience has helped me improve my skill and understand real working environment better. I also thank my lecturer and university for their support and encouragement. To further begin my presentation, we will move on to the first part, which is an introduction to the organization. Arif Effective Sdn Bhd is a subsidiary of All Commerce Industry Sdn Bhd. This company, founded in 2012, Arif Effective Sdn Bhd started off as R&D division of All Commerce Industry, with a consistent innovation in the area of biotechnology and bioprocess engineering. Arif Effective Sdn Bhd amassed a microbial culture of over 600 strains of bacteria and fungi, allowing us to the opportunity to capitalize on this natural microorganism to promote more sustainable agriculture practice. This company organization started with just an R&D department, research and development department that partner with University Technology Malaysia in 2009. The company officially established around 2012 and further worked on many projects throughout year 2013 until 2015. Then, around the year 2017, they are developed a new product called Trico Active Plus to stimulate plant growth and mitigate plant disease. They work on a few new projects for the next few years and finally establish a research and development center at IPAC SAC for microbial research in 2021. Then, in 2022, the installation and commissioning of bioprocess line comprising of two 1,000-liter bioreactor was made by the organization. Next, these are the team members of Arif Effective Sdn Bhd. The head of the organization will be the chairman, which is a Datuk Dr. Abdul Shukur Abdul Rahman. The CEO is a Mr. Wan Azhar bin Mustafa. And next, the COO is a Mr. Charles Tan, and the assistant general manager is the Huang Wei Chang. Further down the organization chart, the R&D manager is a Mr. Muhammad Daniel Azman, and the next is the assistant R&D manager is Mr. Alim bin Lamin, and the QA executive is a Miss Mashita Balkis binti Ijodin, and the research executive is a Miss Huda Adlina binti Badrul Hisham. For the chapter 2, which is the summary of industrial training activity, I learned about the function and operation of a bioreactor, which is a device used to grow microorganisms in a large quantity under controlled conditions. Then, some of the key aspects I learned include how to prepare the production medium and how to transfer culture from a small scale, such as a 70 liters, to a large scale, 700 liters, and how to carry out the fermentation process using a bioreactor. Next, the daily tasks include conducting leaching tests on fertilizer sample to evaluate the rate of nutrient release into a solution over a specific period. After each soaking interval, pH and EC readings were taken to monitor changes in acidity and ion concentration, which can indicate how effectively these nutrients are being released from the fertilizer into the solution. Next, I also carry out an antagonism test between Trichoderma spp tricho and specific pathogen with a senior staff to evaluate the potential of tricho as a biological control agent. The test was conducted on PDA medium using the dual culture method where both microorganisms were placed side by side on a agar plate. Then observation we made to identify zones of growth inhibition can indicate the effectiveness of tricho in suppressing pathogen growth. So these are the some of the example of the result we get after doing the test. So if you can look the Petri agar, the color of the green is a tricho. After that, some side tasks I did throughout my internship program at the company will be labeling the product bottle containing the result from bioreactor cultivation with a sticker, perform inventor calculation, prepare agar medium, and lastly, perform root planting. I also perform quality control to detect the presence of bacteria or fungi in a sample. I also recorded daily rainfall water to determine the amount of rain for the watering purpose. After that, I also measured chlorophyll using SPAT and recorded the height of oil palm seedling, giving two different treatment, which is a treatment control and treatment one. I also collected root sample from oil palm tree for root planting purpose as part of the weekly task. Moving on the next chapter, which is the technical report. The first assignment will be the leaching test on the different fertilizer. The objective of this test is to compare the leaching behavior nutrient content in the leachate and residual fertilizer unreleased nutrient of different fertilizer. The procedure of this test started by placing 7 centimeter diameter strainer on 2 liter flask, then placing fertilizer on strainer according to the volume specified in triplicate. After that, conduct sequential watering according to the rate and intervals. Next, collect leachate and proceed with the testing in accordance with the parameter. And lastly, dry the residual fertilizer in an oven at 60 Celsius overnight and wait the dry mass. Lastly, the NPK content in the residual fertilizer will be tested. This is the trial design used for this test. The testing parameter that was not in this test will be the pH, EC, NPK, total plate count, NPK of residual fertilizer after oven drying at 60 Celsius, and the dry mass of fertilizer residual. The second assignment will be the Bacteria Strain Stale Bank Viability Check project. This is the new strain project aimed at identifying the bacteria morphology present on the surface of Petri dish. I use the quadrant streaking method to observe the confirm the morphology on each Petri dish. Then each box contain different vial types of bacteria strain, and to identify their morphology we refer to the indicator provided by the senior staff to recognize the bacterial shape. These are the some of the photo I took while doing this project. These are the some of the example of the bacteria strain that we need to identify. So for the conclusion, during my industrial training at Arif Efektif I learned many useful skills such as how to use the bioreactor, prepare agar, doing the QC, and handle microbial tests. And also I learned how to work in a real lab, follow safety rules, and work as a team with a staff. For my experience, I gave some suggestions to improve the training program, the company, and myself. I believe this training was very helpful and has prepared me well for my future job in the biotech field. This training also give me a lot of new knowledge and helped me understand how biotechnology company works. It also made me more confident and responsible in doing lab tests. Two suggestions for faculty industrial training program. So the first is the project-based learning. This method allows students to experience real industrial challenge by working on a specific project. For example, they can develop a microbial product or test its effect on soil with a guidance from a company mentor. Students also can learn how to plan, conduct, and report their findings. This can also help them improve their problem solving, lab skill, and confidence in the handling industry tasks. So for the second is the job rotation training. This method also can give students exposure to different roles and tasks within the company. For example, they may first learn microbiology technique in the lab, then move to the quality control to check product standard, and finally experience the production process. With a guidance from mentor in each section, student can learn by doing, not just observing. It can build a full understanding of how the company operates and what each role requires. Next is the two suggestions for industry. The first is accept a simple student lab corner on a training zone. Arif Effective can allocate a small section in the lab as a student zone where intern can carry out simple training tasks like media preparation, sample, planting, or self-handling of culture. This can help students gain practical skills without interfering with the company core operation. It can also give students confidence to work independently and reduce error during their learning process. And the second is the collaboration with university for R&D project. Arif Effective can build a stronger ties with university by collaborating on research and development project. This can include allowing student or lecturer to work on actual company problems such as microbial testing, fertilizer development, or bio-based formulation. This also can help the company gain fresh idea from academic input while giving student valuable real industry experience. It can also support innovation in a product development at a low cost. Two suggestions for myself. The first is take initiative to learn beyond instruction. As a trainer, it's important for me to take initiative. I will not only complete the task given but also show interest in other areas such as fermentation, QC, or microbial testing. By asking questions and observing how the company works, I can understand more than just my assigned role. This proactive attitude will help me gain better experience and show the company that I'm serious about the learning. So for the second is the practice good lab habit and communication. So to be a responsible trainer, I will follow a proper lab practice such as wearing a lab coat, handling samples safely, and cleaning my work area. I will also focus on improving my communication, whether it's asking for help, giving updates to supervisor, or working in a team. This show my professionalism and help me build trust with a company staff. So for the lastly, for the who that later will doing internship, I very recommended to this company Arif Effective to you to learn about the agriculture sustainability practice. So that is all from me and thank you.